Marine Population Genomics Lab at Boston University. And today I'm going to be talking about some of the work that we've been doing on the recent expansion of Acropora hyacinthus, pictured here in the Western North Pacific. So normally during coral talks, you'll start with some beautiful before pictures and some really depressing after pictures like those shown here from American Samoa, where the first and the last picture are just a year apart. And we show these depressing pictures because this is representative of what's happening all over the world due to rising temperatures. But what I'm going to be talking about today is a reef that looked like this kelp bed on the left 40 years ago. And today it looks like this coral reef on the right. So the coral of the day is Acropora hyacinthus. It has a really extensive range, which includes this region in the Western North Pacific. Everywhere in red, you can find these corals. And then since 1980, this coral has also been seen in these additional new blue sites up at the top, all north of where they had previously been found. Well, why did they expand? Well, winter lows have increased by about 1.5 degrees in the last 100 years, especially in the waters surrounding mainland Japan, which I've drawn this blue circle over here. The numbers next to the plus sign indicates the degree increase in the last 100 years, and then these numbers next to the sites, these indicate the current winter lows at each of those sites. So there's been major shifts of habitat in the last 40 years in this region, at least in around mainland Japan. And at one site called Tosa Bay, we have this picture of an Aclonia cava bed in the early 1990s. That same bed in 1997, and then again in 2000, and again in 2013. As you can see, the macroalgae has been completely replaced by coral, by Acropora hyacinthus here. And this habitat change has been attributed to the increased abundance of these two guys here being found more often in this region surrounding mainland Japan, likely due to the rising temperatures that I talked about on the last slide. So the Davies Lab has had a long battle with obtaining permits in this region, which started in 2009 and then ended in 2016. We ended up sampling from 2 sites from the subtropical region here, which I've circled in red. These are samples from the Ryukyus Islands. And then also samples from around mainland Japan, which I've circled in blue, and I'm just going to call this the temperate region. And those samples included corals from one recently expanded site, Amakusa, which on the next slides, that'll be the one that has this little asterisk. And then so for these, we used 2B RADseq. That's the RAD protocol that we use in the lab. We mapped to Acropora mellipora, and then we used ANKs for downstream analyses, and we did our demographic analyses in GADMA. So the first thing that we found was the presence of these 3 different genetic clusters. So we're calling these 3 different lineages, but since this talk is focusing only on the recent expansion, I'm only going to be talking about this single lineage that is found in it's the only lineage that is present in the temperate region here. So I'm only going to be talking about these 5 sites. But if you want to hear what we're doing with these cryptic species, you'll have to reach out to me at some point. Okay, so our objectives here are to 1, confirm the expected direction of expansion south to north. 2, ask if the recently expanded site looks different, and I'll explain what that means in a second, what looks different means. And then 3, if they are different, what do we think is driving these differences? So for the first objective, we use Slatkin-Psi. This takes the unfolded site frequency spectrum and then calculates this psi statistic, which indicates the direction of expansion and then also the strength of the signal of expansion. And the thickness of arrows here, that denotes the strength of the signal. And we see here a really strong signal of expansion from Kushima to Amakusa. Amakusa, again, the recently expanded site. And then also from Kushima and Tatsukushi to these two sites here, Shirahama and Kushimoto, which were present before 1980, but are similar to Amakusa in that they also exist on the range edge. They're further north compared to these more central sites. So the second objective focuses on testing some of these hypotheses that we have about how a recently expanded site acts. And these assumptions include lower effective population size, decreased genetic diversity, increasing LD, and then also I threw increased number of clones here, just because when we have a recently expanded site, those populations are often isolated, and then reproductive strategies can change in more marginal sites. So we use GADMA for the all the demographic analyses, and what we have here is we see low effective population size in Amakusa, and then also low genetic diversity. But we see that it's really the separation between the two more central sites in lighter blue and the three range edge sites in darker blues. LD, there's not a huge difference, but it's still the same general trend where these two central lighter blue sites have lower LD compared to the darker edge sites. And then also we see this similar pattern with the number of clones found. So there's a higher proportion of clone pairs in the three edge populations compared to the two more central sites. So we're seeing a trend with these various metrics from the last few slides, right? There's a separation between the three edge sites and the two central. And it's not recently expanded Amakusa versus everything else. It's edge versus central. And here we just have our pairwise FST plot, and then also this MDS plot, and it's just showing the same thing, the separation between the three edge and the two central sites. So what's driving these differences? Well, it could be entirely neutral, but we know that there's a temperature gradient within this region. Again, these are the winter lows at each site. Those three edge populations experience colder temperatures, like around 15 degrees, and the two more central sites, their winter lows are around 17. So if temperature is important, we would expect to see some evidence of differential selection pressures. So this ratio of theta of non-synonymous over theta of synonymous sites, this is an indicator of purifying selection. So this ratio is lower when purifying selection is higher. And so it looks like these edge sites are experiencing higher purification, purifying selection compared to the two more central. And that might be counterintuitive if you know that the recently expanded sites generally have greater mutation load and so have reduced purifying selection often. But if there's this purifying selection could be caused by more extreme temperatures at the edge sites. It could also just be purging of deleterious alleles due to just from genetic drift. And we know that isolated populations with smaller population sizes are often are subject to higher drift. So it doesn't really eliminate either hypothesis, the neutral versus temperature hypothesis. We would also expect edge sites to show different physiological responses to temperature from the central sites if temperature is driving these differences. So we have these really sweet respiration chambers that we can use to measure dark respiration and photosynthesis, and we plan to measure, we measure these across different temperatures to get an idea of the thermal performance curve of the organism. And ideally, we would do this at each site with a bunch of samples from each site. That's been rain checked due to travel restrictions. But we would also expect a genomic signature relating to temperature tolerance. And for that, we've done Bayescan and PC adapt, just doing the edge versus the central sites, and then taking the outlier loci that, that appear from both Bayescan and PC adapt. And then we look plus or minus 1 kb of the loci for nearby genes. And then I just took this list of genes from Dixon et al. 2020, which compiled these different gene modules, looking at multiple experiments where they stressed out acroporids and looked at the genes that were differentially expressed. So we found of our 6 genes that were near outlier synapse, 3 of those were in these modules, including this peroxidase gene. So just a quick summary here, we found a lower effective population size and a heterozygosity, higher LD and more clones at the edge populations. There's a separation between edge and center populations. The edge seems to be experiencing higher purifying selection. There's a possible genomic signature linked to temperature tolerance from the outlier loci analysis. But most importantly, we need more samples to do the physiological stuff. And with that, I just want to thank all of the people that have helped with this project.